Hello, everyone. Welcome to my video tutorial for Bisulfite sequencing data analysis. In today's video tutorial, I'm going to show you how to examine the methylation level within the gene promoter regions. Because the DNA methylation in the gene promoter regions is an important biological process to regulate gene expression. So I'm going to use the same dataset, GSE 86297, for today's demonstration. In my previous video tutorial, I showed you how to download the data from NCBI website, and I explained the data in details in that video. So today we are going to the same process to load the data. First, we can load the target files. Let's run. We can have a look at the target files again. You can see we have 6 files in this dataset. Here are the files name that we downloaded from NCBI website. You can see in this dataset we have 3 groups according to the oversight size. Group 40 micrometers, 50 micrometers, and 60 micrometers. Each group has 2 repeats, sample A and B. So now we can use the file's name in the target to create a DGE list. Again, we are going to use the read Bismarck to DGE function. Because for all 6 data samples, the cytosine positions were quantified by the Bismarck package. So the files will be the files name in the target, and the sample names will be the samples name in the target. So let's create the DG list from the bisulfite sequencing data. You can see R is reading all 6 samples. Now we have a DG list data in our data window, and we named it as Y4. So again, we are not going to analyze the mitochondria DNA, so we can remove the mitochondria DNA. Let's run. We can use the table function to check if we remove the cytosine positions in the mitochondria DNA or not. Let's run. You can see the count for the cytosine positions on the mitochondria DNA is 0. So next we can reward the chromosomes from chromosome 1 to 19, then X and Y. So let's reward the chromosomes. And we can use the table function to look how many cytosines in each chromosome. You can see now the chromosome in correct order from 1 to 19, then X and Y. Because those samples are from mouse oversight, so we know mouse has 20 chromosomes. And in the second row are the total numbers of cytosines on each chromosome. Because we want to analyze the DNA methylation in the gene promoter region, so next we can annotate the CpG locus with the identity of the nearest genes. We can use the nearest transcription start site function to do the annotation. Let's run this function. You can see we created a new data frame named as TSS. We can click this data frame and have a look at the data information inside this data frame. You can see we have the first column as gene ID, second column as gene symbols, and the gene weights. Here is the transcription data set for each gene. then the strand information, and the distance for the nearest cytosine. You see the distance is in minus. That means the distance for this cytosine is downstream of the gene promoter XKR4 from the transcription starter site. So now we can add the gene ID, gene symbols, with strand and distance information into the DGNIST. So let's add those TSS information into DGNIST. First is the gene ID, next is the gene symbol, then gene weight and the strand information, and the last one is the distance. So we can have a look at the gene data slot in the DGNIST. So you can see now we have the chromosome information locus for each cytosine, and also we have the nearest gene ID, gene symbol, weight, strand, and distance information. So we got the data ready to analyze the DNA methylation in gene promoter regions. So first we need to define a region for the gene promoters. In this demonstration, we are going to set the gene promoter region as downstream 1000 base pair and upstream 2000 base pair from the transcription starter site. You can change the numbers to set the gene promoter regions for the genes that we identified in this dataset. So let's set the promoter region first. Then we can have a look at how many cytosines positions are located in the gene promoter region that we set it for all the genes in this dataset. We can use the table function. You can see we have nearly 800,000 cytosines were located in the gene promoter region. So now we can subset the data just to keep the cytosines that are located in the gene promoter region. We can subset the data now. If we have a look at the gene promoter region DGNIST, you can see now we only have 797,131 cytosines from our 6 samples. I showed you before that we have 12 columns here because we have methylated and unmethylated cytosine positions for each sample. So next we can summarize the total count for each gene promoter within each samples. We can use the row sum function to do this. Let's run, and we named it as Y promoter now. So let's get the count from the Y promoter, and we can click the count and have a look. You can see the row names are the gene ID. We have 21,838 gene promoters. And here are the total count for the methylated and unmethylated cytosines in the gene promoter region. So next we can perform the data filtering. Again, we are going to do the data filtering as previous demonstration. First, we add the unmethylated and methylated cytosines numbers together for each sample. So let's label the data columns for methylated and unmethylated columns for each sample. Then we can get the methylated and unmethylated count for each sample, and we can add the methylated and unmethylated count together. So now we have the total numbers in each gene promoter region. So now we can set a threshold Again, we are going to use 8 as the threshold to filter the gene promoters for all 6 samples. So let's label how many gene promoters have cytosines more than 8 numbers. And also we want to remove the cytosines that always show methylation or never been methylated, because those cytosines not going to give us information for differential methylation in the promoter regions. So we can label those cytosines as well. Now we can use the table function to see how many cytosines meet our requirement. You can see now we only have 15,000 and 44 gene promoter regions that have count more than 8 cytosines, and all the cytosines could be used to perform differential methylation analysis. So now we can subset the data again. Just keep the 15,000 and 44 gene promoter regions. Let's subset the data. If we have a look at the Y promoter DG list again, you can see now we only have 15,000 and 44 gene promoter regions. So in standard agile workflow, after the data filtration, we normally perform data normalization. But for DNA methylation analysis, we don't need to perform data normalization. However, we need to set the library size for each sample to be the average of the total read count for the methylated and unmethylated libraries. Before we do that, we can have a look at the samples from the dataset. Let's run. You can see at the moment the library size is different for methylated column and unmethylated column for each sample set. Generally, we can say there are more unmethylated cytosines than methylated cytosines in each sample. So we are going to use the average library size for the methylated and the unmethylated library size. So let's first get the average library size for each sample. Then we can set the average library size for each sample. If we have a look at the data list for the samples in the DG list again, you can see now the library size is the same for methylated and unmethylated column for each sample. So we set the library size equal between unmethylated and methylated column for each sample. Next, we can design the matrix to perform the differential methylation analysis. First, we can design the model matrix to include the group information, because we use the 0 plus here, so we will include all 3 groups. Let's run. We can have a look at the matrix design. You can see we have 3 groups: group 40 micrometers, group 50, and group 60 micrometers. But for DNA methylation analysis, we need to expand the matrix design to modulate the sample and the methylation effect. So there's a function in HR called the module matrix methylation. This function is specifically designed to expand the matrix design for DNA methylation analysis. So now we can use this function to expand the matrix design. If we have a look at the matrix design now, you can see we have 6 columns from column 1 to column 6 to show the sample information, sample 1 to sample 6. And we have the last 3 columns represent the methylation levels in 3 different groups. So we designed the correct matrix now. Next, we can estimate the dispersion. Once again, we don't estimate the trended dispersion for DNA methylation analysis, so we can set the trend as none. Now we are ready to estimate the dispersion. So after the dispersion, we can fit the generalized linear models for the data. Following fitted model, we can perform differential methylation analysis. Because we have three groups here, we are going to compare group 6 micrometers against group 40 micrometers. This is the size for the lower size in culture. So first we can use the make contrast function to set the group comparison for 60 against 40. Let's make the contrast. Now we can use the contrast to perform the likelihood ratio test. So after the test, we can use the decide test to summarize the gene promoter regions that show up or down methylation in group 6 against group 40. We can summarize the data. You can see here is the analysis that we compare the DNA differential methylation between group 60 and group 40 micrometers. You can see we have 15 gene promoter regions that show decreased DNA methylation and 697 gene promoters regions that show increased DNA methylation in the 60 micrometers group against the 40 micrometers group. So now we can make a plot to show the summarized data. Before we do that, we can expand our plot windows. Now we can make the plot. You can see we generated the plot for the summarized data. Here are the gene promoter regions. Red dot show the gene promoter regions that have increased the DNA methylation, and the blue dot show the gene promoter regions that have decreased the DNA methylation in the group 60 micrometers against group 40 micrometers. So we know the numbers for up and down regulation. We can also use the top text function to look at the top gene promoter regions. For example, we can set the n number as 20, then we can look at the top 20 gene promoter regions. You can see here are the top 20 gene promoter regions that show upregulation. We have the gene IDs, the row names, we have the chromosome information, gene ID, gene symbol, strand, log FC changes, log CPM count per minute. And down here we have the p-values and the values for false discovery rate. So we only show 20 top regulated gene promoter regions. You know from the data we analyzed the 15,044 gene promoter regions in this dataset. We can get all the information we showed here for all the gene promoter regions. If we set the n number as 15,044, then we can get all the information for all the gene promoters. You can see we have a large top text data frame here. We can click and have a look. You can see all the information that we want are in the table data slot. So we can get the data from the table data slot. The data frame, we can name it as differential methylation. You can see in our data windows, we have a data frame DM. Let's click it. You can see we have all the information for each gene promoter regions. We summarize the significant changes for the DNA methylation. Use the first discovery rate as 0.05, so we can filter the data to just keep the significantly up and down-regulated gene promoter regions. So let's filter it. You see now we only have 712 gene promoter regions. Those numbers include the 15 downregulated gene promoter regions and 697 gene promoter regions. So if you want, you can save the data frame as a CSV file. Then you can open it in your computer. Let's run. So we saved the data file for our analysis. Okay, that's it for today's demonstration. If you want to analyze differential methylation in gene promoter regions, you can follow my video tutorial to analyze your dataset. I hope this video can help your data analysis. If you have any questions or queries, you can leave your comments in my video channel.